The overall goal of this procedure is to quantify CpG methylation in targeted regions of interest out of genomic DNA isolated from tissue or cells of interest. This is accomplished by first designing bisulfite-specific PCR primers to amplify regions of interest from bisulfite-converted genomic DNA. In the second step, the bisulfite-converted region of interest is amplified and next-generation sequencing, or NGS, libraries are generated from the amplicons. Next, the libraries are sequenced and FASTQ files are generated for analysis. In the final step, the sequencing reads are aligned to the bisulfite-converted reference target sequence and the 5mc% is calculated. Ultimately, bisulfite amplicon sequencing, or BSAS, can be performed to precisely and accurately quantify the CpG methylation in any region of interest from any model organism.
The main advantage of this technique over existing methods, like Sanger sequencing, is that BSAS provides accurate and precise CpG methylation quantitation in a rapid, high-throughput manner, providing the opportunity to assay multiple regions in a high number of samples.
After identifying the target and designing the primer, assemble the reaction for the target amplification optimization of a single amplicon, and then seal the PCR plate with heat-sealed film. To visualize the amplicons by page electrophoresis, next load the appropriate wells with 30 microliters of PCR reactions or ladder, and then run the gel at 200 volts for approximately 45 minutes. When the first dye reaches the end of the gel, stop the reaction and then cover the entire gel with 5 micrograms per milliliter of freshly prepared ethidium bromide. After 5 minutes, image the gel via a UV transilluminator at an excitation wavelength of 482 nanometers. Use the ladder to size the PCR amplicons. To determine specificity of the reaction, look for multiple bands other than the expected amplicon size. To determine the average size of the detected library peaks and an estimation of the molarity, next run the libraries on a capillary electrophoresis DNA sizing chip with a high-sensitivity DNA assay according to the manufacturer's instructions. To quantify the libraries by qPCR, use primers designed against the adapter sequences in the generated libraries and standards with a known concentration on a real-time instrument using the absolute quantification settings according to manufacturer's instructions. Then use the size of the libraries and the molar quantitation from the qPCR to calculate the molar concentrations of the libraries. After sequencing, import the zipped FASTQ files into the appropriate sequencing analysis pipeline. Select Import, and then select the sequencing method used to generate the reads. To retain the Q-scores for the read trimming applications, import the appropriate zipped FASTQ read files, and then under General Options, deselect Discard Quality Scores. Select a location for the imported reads and click Finish. Then under NGS Core Tools, select Trim Sequences. Highlight the reads to be trimmed, retaining only the reads containing greater than or equal to Q30 scores. Then select the Trim Using Quality Scores and set the limit to 0.001. To further select only the reads containing less than or equal to 1 ambiguous nucleotide, select Trim Ambiguous Nucleotides and set the maximum number of ambiguities to 1. Then select a location to save the trimmed reads and click Finish. To map the trimmed reads to the in silico bisulfite converted gene reference, under NGS Core Tools, select Map Reads to Reference. Select the trimmed reads to be mapped and click Next. Then, under Reference Masking, select the converted reference sequence and select No Masking. Click Next, and then assign a score of 3 for mismatches, insertions, and deletions, assign a score of 1 for the minimum fraction of read length mapped to the reference, and assign a score of 0.9 for the minimum fraction of identity between the mapped read and reference. Under Nonspecific Match Handling, select Ignore and click Next. Under Output Options, next select Create Standalone Read Mappings. Then under Result Handling, click Save. Select a location to save the read mapping and click Finish. To run the variant calling on the mapped reads, under Resequencing Analysis, select the Low Frequency Variant Detection and choose the read mapping to be analyzed. Click Next, and then under Low Frequency Variant Parameter, enter 0.01% and click Next. Under General Filters, set the minimum coverage to 1000 to ensure that the sequence is at a depth of greater than or equal to 1000x. Finally, click Next, and then under Output Options, select Create Annotated Table and select a location to save the variant table file. Click Finish and use the variant frequencies at the annotated CPG sites in the region of interest from the variant table file to calculate the frequency of the cytosine methylation at a reference cytosine in CG context. Bisulfite amplicon sequencing reads properly aligned to the converted reference sequence will resemble those illustrated in the figure. The CPG dinucleotides can clearly be identified, and the methylation states can be estimated by observing the base calls at the CPG sites in the mapped reads. 0% methylation will result in mapped reads containing thymines mapped to the CPG sites. 100% methylation controls will result in mapped reads containing cytosines mapped to CPG sites. In this experiment, RNA and DNA were co-isolated from a mouse cerebellum and retina. The rhodopsin expression, selectively expressed in retinal tissue, was then measured using qPCR. As expected, the rhodopsin RNA was detected only in the retina. After quantification of the CPG methylation levels in the rhodopsin promoter region by bisulfite amplification sequencing, however, the cumulative methylation levels across the promoter region were greater than 80% in the cerebellum compared to less than 15% in the retina. BSAS methylation quantitation benefits from site-specific methylation quantitation, allowing the comparison of the methylation levels on a CPG-specific basis across any given genomic region. Indeed, in this experiment, the CPG methylation levels across the rhodopsin promoter were found to be significantly higher in the cerebellar samples when compared to the retina samples.
After watching this video, you should have a good understanding of how to quantify CPG methylation in targeted regions of interest out of genomic DNA isolated from either tissues or cells.